Hello everyone, let's learn about UV visible spectroscopy. So first of all, what is UV visible spectroscopy? It's a technique used to analyze the absorption, transmission and the reflection of ultraviolet and visible light molecules by molecules. Next one is a principle. The basic principle of UV visible spectroscopy involves the absorption of ultraviolet and visible light by molecules. When the molecules absorbs light, which causes the excitation of electrons from lower energy level to higher energy level. So this absorption of light by a molecule is given by Beer-Lambert's law. This Beer-Lambert's law, it's a combination of two laws that is given by Johann Lambert and August Beer. So hence it's known as Beer-Lambert's law. When a monochromatic light, that is a single color of light, initial intensity passes through a solution in a transparent medium. So some of the light get absorbed and some light gets reflected. So Beer-Lambert's law states that the concentration of a solution for the non-path length is directly proportional to its absorbance of light. A is equal to epsilon Cl. Here A is the absorbance, C is the concentration of solution in mole and L is the path length of sample. Next one is types of electron transition. There are 4 types. First one is sigma to sigma star transition. In this transition, the electrons get the electrons in a molecules will move from bonding to anti-bonding orbital. This takes place due to electromagnetic radiation, and it requires a high quantity of energy, and this can be observed in methane molecules due to the presence of only carbon hydrogen bond. And the second is N sigma star transition. In this transition, the electrons from a non-bonding orbit moves to an anti-bonding orbit. The lowest quantity of energy is required. Example, halogen and elements like sulfur, oxygen, nitrogen display this kind of transition. Third one is N pi star transition. In this transition, the electrons of non-bonding orbital moves to an anti-bonding orbit. So compounds containing double bond involves heteroatoms that undergo this kinds of transmission. And the last one is pi to pi star transition. In this transition, the electrons move from a bonding orbital to an anti-bonding orbital. The common examples are the aromatic ones like alkenes, alkynes, nitriles and carbonyls. And an illustration, here is the illustration of different kinds of transition. So the illustration in which the energy is represented in the x-axis and orbitals are arranged in the order like we can see here. The arrangement starts with the sigma to sigma star bond at the lowest, then to n to sigma star, then n to pi star, then last one is pi to pi star. And in spectroscopy, there are 2 types of beam. First one is single beam spectroscopy. Here in this single beam, the light waves, it will pass through the sample. So here the inside the cuvette sample is present, so this light source will pass through that sample. And in double beam, this light will pass or will split into 2 parts. One part will passes through the reference and the other will passes through the sample. Next one is instrumentation. The main part of the UV spectroscopy includes light source, wavelength selector, sample containers, detectors, amplifiers and recording device. So light source, which is the most essential component required for emitting the light in a wide range of wavelength. So to work this spectroscopy, we need a wide range of wavelength. So commonly light sources that have high intensity will be used, such as deuterium lamp that is used for UV light, and halogen or tungsten lamp for visible light as a source is used. The second one is wavelength selector. The commonly used wavelength selector in this UV spectrophotoscopy UV spectrometer is monochromator. So this monochromator, it will convert a polychromatic light into a monochromatic light. From the entrance slit, light of different wavelengths enters into this monochromator, and at a particular angle this beam will collide and it strikes the dispersing elements. And the monochromator, it contains a prism. This prism, it will separate all the different wavelength of light into a single beam, and only a single radiation or a color of a specific wavelength will be allowed to leave the monochromator, and it will ultimately pass through the exit slit. A chopper will be present, so which helps to split the monochromatic light into two beams, one that will passes through the sample while the other through the reference, like in double beam spectroscopy. Third one is sample container or cells. So the sample and the reference solution must contain a radiation transparent cuvette. This will be made of glass, quartz or silica cuvettes. So through that cuvette the radiation will pass. The fourth one is the detector, a device which converts the light energy into electrical energy, and that will be displayed on a readout device. Sample absorbs a part of radiation and the remaining will be transmitted. This transmitted radiation will be this transmitted radiation will fall on the detector, which determines the intensity of the radiation. The higher the current, greater is the intensity. It has the properties of low noise and high sensitivity, so it gives a linear response. Each detector has a variety of wavelength ranges and different sensitivity. Finally, the data recorder usually plots the absorbance against a wavelength in the UV and visible section of the electromagnetic spectrum. And there are types of detectors will be used. First one is photomultiplier tube, second is photovoltaic cell, and third is phototubes. Fifth one is amplifier. This amplifier amplifies the signals from the detector many times to produce a clear and a readable signal. Sixth is recording device. It records all the data and produces the spectrum of desired compounds. So this is a basic instrumentation of a spectrophotometer. Light source, a beam of light will enter. This monochromator has prism, so this light get reflected, and single beam of monochromatic light will be passed through this sample solution, and this passes through the detector, and digital display will be observed at last. Application. This that is UV visible spectroscopy used in cosmetic industry to identify the photostability index, quantify the dyes and antioxidant, and to detect the adulteration. This can be also used in material science for the like characterization of nanoparticles, and to determine the battery composition. Used in cancer research to estimate the hemoglobin concentration. Also used in biochemistry and genetic field, used to quantify DNA, protein or enzyme. Also used in petrochemistry for characterizing crude oil and the quality of the crude oil gravity. Thank you.